Hello, in this video we will discuss about the biochemical effect of chronic alcohol consumption. So the chronic alcohol consumption as compared to the normal consumption of the alcohol is very dangerous as the chronic alcohol consumption. So the all metabolic path of alcohol breakdown occur in the liver cells. So remember about that, and hepatocyte, the known of the cell of the liver. So basically here is the alcohol when we will intake go into the stomach. So first of all go into the stomach and the water absorbed from the digestive system to into the digestive system first. So it will go into the duodenum, and here is the cell is known as the brush border enzyme containing cell is known as the enterocyte. So these enterocytes basically is surrounded by the basically is the mucous membrane is surrounded by the enterocyte, which that is used for the barrier immunity and contain a microbiome. This microbiome or microbiota contain a gram-negative bacteria which that consume alcohol and produce the basically lipopolysaccharide, and this lipopolysaccharide go into the blood, and increase the lipopolysaccharide will cause the inflammation. So also the dehydration of the tissue will be caused due to the inside movement into the blood. So alcohol will move into the blood, so it means absorption, and that means the water will be dehydrated from the tissue to go into the blood and increase the water inside of the blood to increase the blood pressure. So on the other hand, here I am drawing the liver. So this alcohol which that is present in the circulatory system go into the liver in the hepatocyte. So let's begin to understand the hepatocyte and what is the liver containing things. So the liver is contain lobules. These lobules contain a sinusoid of liver. So here is the sinusoid of liver. This cross section of the lobule shows that the sinusoid of the liver which contain a Kupffer cell, and the Kupffer cell is basically important part of the cell which that is recognized is looking like a basically is the Langerhans cell like structure. So here is the hepatic the dendritic cell like nature. So here is the hepatic stellate cell which that is present. So when the lipopolysaccharide recognized by the toll-like receptor are present in the Kupffer cell, and it will cause the positive effect to the hepatic stellate cell as well as cause the inflammation and produce the inflammatory mediators to cause the neutrophil and macrophage recruitment. So that's cause the inflammation and the cause also the fatty liver by the production of the collagen. We will discuss in another video with the detail, but here is just we will understand the biochemical pathway of the chronic alcohol consumption. So when go into the hepatocyte, so here is I am drawing the whole picture of the hepatocyte contain a plasma membrane. Thus plasma membrane contain a GLUT4 channel for example, and move the glucose inside from the outside, for example into the blood circulatory system to the inside of the hepatocyte. So here is let's begin to understand first the effect of the glucose on alcohol, and their basically is the relation alcohol and sugar which that is we will also discuss. So the alcohol diffuse directly into the plasma membrane, so it cross the barrier, and it is basically ethanol is convert into the acetaldehyde. It is the 75% the reaction is the aldehyde dehydrogenase enzyme. Ethanol acetaldehyde dehydrogenase enzyme is used. So basically is the produce NADH in this case. While on the other hand here is the another mechanism. 25% reaction are present in the cytochrome pathway, which that is lead to use the NADPH and produce the water and also produce the reactive oxygen species in the presence of oxygen. So that is basically is the organelle which that is used for it. So here is we will discuss about the reactive oxygen species contain basically is the free radical. This free radical is damage the DNA in the nucleus, and this damage to cause the point mutation, addiction of the alcohol, and DNA methylation and epigenetic modification, which that is lead to alter the gene, and their chance can be 25% due to the they depend on the chemical reaction. So on the other hand, here is the production, and that was the cytosol. So here is the mitochondria. Inside of the mitochondria we will discuss. So the acetaldehyde is the aldo sugar. This aldo sugar will inhibit the other glucose incoming inside of the cell for metabolism. So here is the ketoacidosis will be occur when this in the mitochondria converting the acetate, and acetate move into the blood to cause the ketoacidosis. So the acetate or acetic acid production. While on the other hand, the NAD plus reduce into the NADH, and coenzyme synthase enzyme used to produce acetyl CoA, and these two molecule of acetyl CoA will lead to produce the acetaldehyde. So the acetoacetate, and the NADH is increase, and it means the increase of the NADH is used for it, and the acetoacetate will be converted by the lipogenesis to produce the fatty acid, and it is increase the fatty acid means the increase of the cholesterol, because the fatty acid is lead to produce the cholesterol. So the hypercholesteremia due to the HMG CoA enzyme used in the liver, while the triglycerides is also increased due to the increase of the fatty acid and glycerol, and that is the adipose or lipogenesis will be occur. It means the obesity is due to the chronic alcohol consumption. So that is why the people will obese. And on the other hand, here is the NADH increase. So when NADH increase to lead to more adipose lipogenesis, because it is no need to more NADH, it means the increase of the NADH. So the triacylglycerol will move into the liver and increase the low density lipoprotein, while decrease the high density lipoprotein. On the other hand, that increase the cholesterol synthesis due to the triacylglycerol excess. So on the other hand, what is the effect on glucose metabolism? So here is the glucose will enter. So here is the acetaldehyde is the aldo sugar will inhibit the metabolism of the glucose. So it means the hyperglycemia will be occurring in the blood circulatory system due to the excess of the glucose in the blood circulatory system, while inhibit the glycogenolysis. So it means the glycogenesis will be occur, and after the glycogenesis the hypoglycemia in the blood, a low blood sugar will be in cause. So that is both symptom can be. On the other hand, here is the glycolysis, which that is lead to produce 2-pyruvate. So this 2-pyruvate is the oxygen converted into carbon dioxide. So that is the aerobic respiration, which that lead to enter into convert acetyl CoA, and this acetyl CoA go into the Krebs cycle to produce carbon dioxide, ATP, and NADH. While ATP is we will taking from the acetyl CoA, which that is come from the alcohol. So we will not need to consume the sugar as compared to the alcohol, because alcohol is giving the acetyl CoA, and this acetyl CoA is basically is present in the mitochondria, which that is come from the alcohol, and that's produce the ATP, and this ATP is inhibit the ATP is come from the glucose. So the glucose inhibition of the catabolism of the glucose will be inhibition. So the glucose catabolism inhibition and alcohol metabolism run. So that is the all overview about the biochemical pathway of alcohol. Thank